Welcome to the Science Fair 2020. This year we have our young scientists presenting more than 80 projects. We are having so much fun here. Come join us.
My project is about how do we optimize the setup of renewable energy to maximize its production.
Dr. Bunhead, and as you can see, there's a whole pile of kids here. We've just had an explosive, pure live science show.
An amazing experience, a lot of happy kids, a lot of people enjoy for seeing science live. It's not magic, it's science, and it's awesome.
Good evening, everyone. I'm Kevin Cullen, Vice President for Innovation and Economic Development here at KAUST. And it's my great pleasure to introduce Professor Mark Kendall, a fellow Australian from the Australian National University in Canberra, a university I know well because that's where my youngest son is currently studying. Professor Kendall is an internationally recognised innovator and researcher who's worked at the University of Oxford, the University of Queensland, and is currently the Vice-Chancellor's Entrepreneurial Professor at ANU. There he's the founder and CEO of WEP2020, a company created by ANU with the explicit intention of doing research that will lead to commercialisable outcomes in the healthcare industry. In my line of work, it's fabulous to see a university being innovative and entrepreneurial in this sort of way and driving great research to deliver public benefit. It's a risky approach for a university, but I think it's a calculated risk on the part of ANU, as Mark has over his career developed innovations which have generated $2 billion in value in the healthcare sector. And I believe he has only one KPI in his current job, and he said that's to succeed. And that's a reasonable KPI to have. But to prove that he's not only a great scientist and entrepreneur, but a real all-rounder, and that's a cricketing term, Mark actually holds the cricket bowling record at Magdalen College in Oxford, a cricket club that's been around for 350 years. I'm not a cricketer, but I believe that his record of 6 wickets for 80 runs is almost as impressive as his scientific record. It's my absolute pleasure to introduce Mark Kendall, and please join me in welcoming him to the stage.
Thank you. Thank you. So it's a real pleasure to be here today, and I'd like to thank the organisers for inviting me to come across to Saudi for the first time and to be here at KAUST. It's a complete pleasure. So I'm going to try and do something a little bit different today. We're going to run a live experiment, and I'll explain what that looks like in a moment. And it's a test with our micro-wearable device. Now, I'll set a little bit of context on this. The goal of my talk today is I look around in this audience. I see a lot of people who are scientists doing PhDs or postdocs, and the scientists and engineers primarily. And what I'd like to do is encourage you to field hop, if you can. And also, I know 5-year plans are a thing or a fashion these days, but I encourage you also to sometimes step away from that, because there's opportunities that will hit you across different fields, especially with what's coming with the 4th Industrial Revolution. And I encourage you to embrace that. So I'll explain what I mean by that through this presentation. So first, a little bit of context, some ground rules, if you will. So what is the 4th Industrial Revolution? So let's have a look at what the others were and how that played out. Now, it's tempting to show you a technology. I love technologies. I love medical devices. We'll talk about that in a moment. But we'll do it a little bit differently. It's the human story behind it. So let's start with this picture here. This is in the 1850s, and this is from the First Industrial Revolution. And this guy is one of my heroes. His name's Isambard Kingdom Brunel. And he is, if not one of the champions of the First Industrial Revolution. Now, the First Industrial Revolution was steam power, mechanized work, things like that. And it changed the face of the world. It changed England, it changed the world. And I just love this picture. It was just before a massive ship called the Great Eastern set sail, and they're the chains on that ship. That's the first industrial revolution. What about the second? Well, I might do a quick quiz, if I'm allowed to, and ask anyone in the audience, can you recognize who this individual is? Feel free to say yes. Thomas Edison. Well done. So, Edison's a champion of the second industrial revolution. And the second industrial revolution is things like electricity, mechanization, and also mass production. And the era for this, experts in this area suggest, was about the 1870s through to about the 1950s or so. It was faster than the first industrial revolution, and people like Edison have changed our world. And that's fantastic. Now, I don't need to do a quiz for this guy. I think we all know who he is. And so Steve Jobs is a champion of the third industrial revolution. And the third industrial revolution has happened pretty much in our lifetimes. So what is it? It's computers, it's the internet. And indeed, many of you will be holding something in your pocket, which is an embodiment of the third industrial revolution, which is an iPhone or something like that. Now, if we look at the third versus the others, the third was faster and its impact has been even greater, and it's still playing out in front of our eyes. So what is the fourth industrial revolution? I've given a blank screen on this. So the fourth is, it's a complexity. So the fourth is there's a whole bunch of things taking off at the same time, okay? So let's start with the thread I've just touched upon, which is the computers and the internet and so on. So that trajectory is still exploding in front of our eyes. It's estimated, with the work I'm doing with the World Economic Forum, that within this decade that we're in, the number of devices connected to the Internet of Things, if you will exceed 1 trillion devices, and probably in about 4 or 5 years from now. So that's just one thread, but the point I'm making is there's a bunch of other threads as well. So we have genomics, we have phenomics, we have robotics. There's all manner of other things taking place, all of them exponentially taking off. Now, what's the fourth industrial revolution is that it's those things being put together and integrated. Imagine like the third here as an iPhone, and I don't have any shares in Apple. I'm not advertising Apple, but that's an integrated device that's spanning a few different things. Well, it's quite possible with the fourth industrial revolution, there's devices that we can't even imagine because there's so many different technologies taking off at the same time. That's what it is. It's estimated it's going to be faster than ever before. That's believable, and its scope and its impact will be greater than anything that we've ever seen before. Jobs will change. So just as one example, in 1990, the 3 biggest manufacturers on the planet, the 3 biggest companies in valuation, were all centered in Detroit. So it was Ford, General Motors, and Chrysler. And they employed about 2 million people. Within 15 years, that fell away very quickly. And the top 3 are all in Silicon Valley: Facebook, Google, and the other one a little bit further north, Amazon. And they only employ about 200,000 people. So there's a lot of changes that are already happening with the third. It's estimated the fourth, it will take off. So what's this got to do with healthcare? So I'll switch gear for a moment and show you another historical picture. Now, before I go into too much detail, I just want to say one other thing about the fourth industrial revolution. Your generation, the people I'm looking at in this room, will be front and center in that, and we don't know who the champions of the fourth industrial revolution will be. It could possibly be one of the people here in the room. And wouldn't it be great that it's not even, so there's some problems with the images I've shown, it's all men in black and white pictures. Wouldn't it be great if it's a color picture of someone who's female doing that? So who knows what will happen? That's exciting. But let's switch gear into medicine. So this is a picture, a schematic of the original needle and syringe. It was from 1853, about the same time as that photograph with Brunel, with the chains, right? And that is the primary delivery device for most drugs today, and indeed vaccines. A technology that's 160 years old now, more. and you look at it, it hasn't changed much in how things work. So my journey, I'm going to walk you through my journey a little bit, and the field hopping associated with that has been looking to challenge this paradigm, the flaws associated with the needle and syringe, whether it's delivery or indeed blood draws for diagnostics. Indeed, I'll talk about the fourth industrial revolution, how some of this is playing out. So first, a little bit of backstory and context. My undergraduate degree is mechanical engineering, and my PhD was in a field called hypervelocity aerodynamics. It was in rockets. And I never imagined in my lifetime I'd work in healthcare. And the backstory is, more than 20 years ago now, I was lined up to go to a place called Caltech, which many of you, or all of you, would have heard of, to go and work on rockets, to do a postdoc there. And it was my dream. I love rockets, and indeed, Caltech's one of the center points for rocketry. It's a fantastic place. But then a 2-minute conversation just changed everything. So I was doing something like what I'm doing now. I was giving a presentation at a conference, and afterwards, someone came up to me and said, "I enjoyed your presentation. I have this idea to use rockets to fire vaccines into the skin. Would you like to come and work with me?" I thought, that really sounds interesting. So I asked 2 questions. So the first was, where is this work? And he said at Oxford University. And I asked the second question, which was, would I get a chance to row, as in rowing? And he said, if you're good enough. And I said, okay. Of course, it was a little bit more complicated than that. But within 6 months, I was at the University of Oxford. And what I was asked to do was to turn this device into a reality. Now, this is an example of it in the flesh. It's like a real-life Star Trek device. And the idea was to fire vaccines into the skin at 1,500 miles per hour, in metric about 2,000 kilometers per hour, maybe more, and go very shallowly into the skin and as an improved form of delivery. So there was an amazing adventure being over there at Oxford. I was a youngish lecturer and a rocket scientist looking to do something a bit different. Instead of using rockets to basically do other things like put things into space, or indeed rockets can be killing machines, turn it around and actually come up with applying rocketry in a domain that is designed to keep people alive. So put my shoulder to the wheel, working with others, and indeed Brian Bellhouse was my professor there, and he was a mentor of mine. Set about turning this into real life, and I won't go into all the inner workings of this and how it happened, but in summary, the energy source is helium, 6 megapascals of helium. You press a button, it opens up a canister, sets up a very high-speed gas flow, which accelerates particles within a little cassette, and they impact the skin. And I was really excited about that work. And our job was, let me back up, my job in particular was to make the rocket work. Now, what does it mean by working? Well, you guys are a sophisticated audience. You know that usually it's not just one parameter. There's a whole bunch of different things that need to take place for something to work. And one of the things that I needed to do was to do this, set up high-speed flows of microparticles where all the particles are hitting the skin at the same speed. And if they're able to do that, then the idea was that particles would be controlled and go into the skin to the same depth. Seems to make sense, right? Not quite. We did that. So that red color there to the right, that means they've all accelerated, and at the exit of the nozzle, they're all doing the same speed, in this case about 400 meters per second. But then biology creeps in. Now, I don't want to put you off, but the first moment an engineer looks and touches biology, for me it was a confronting experience because I was blown away by all these sources of variability. And here's just one example. That device I was mentioning before, this is a histology sample of the skin. That's a fancy term for slicing the skin up into thin sections and taking a look at it with some stains, okay? And there you can see the microparticles, some of them on the surface, some of them deeper, and I was thinking, how does that work? I was doing the job and everything was, all the particles are hitting the skin at the same velocity. One of the humbling experiences in this was it turned out the hard part wasn't the rocket. The hard part was the biology. Biology is far more complicated than the rocket science. Us engineers, especially with fluid mechanics background, we're used to seeing things in a tightly defined tolerances, plus or minus a few percent, that kind of thing. And then suddenly, the skin, the biology of the skin is unbelievable. And so we started learning the mechanical properties of the skin. And I started realizing that the sources of variability in the biology is something we need to understand in order to make these devices work. So we started getting into dermatology and measuring mechanical properties. And then on top of that, there was the immunology. So I started thinking, actually the main job here is these microparticles to interface with the skin, a particular type of cell that's important for vaccines and immune function. They're called antigen-presenting cells. I thought, surely there must be a better way of doing this than blasting them at 1,500 miles an hour and then them coming to rest. So let's just pause that for a moment and talk about a commercial picture. The bigger picture is the Gene Gun became Oxford's most successful spin-off company at the time. It was sold for $1.3 billion in 2003. And it was an amazing adventure of learning how to patent things, take them forward, work in interdisciplinary teams and ask questions. And Oxford was really pioneering that environment. But along came Bill Gates, not to me personally, but Bill put out a call in 2003, a call to arms for engineers to try and help improve the way vaccines work. It was called the Grand Challenges for the developing world, improving vaccines for the developing world. So I sat down and tried to make this work for the developing world, the Gene Gun. And I really, really tried to make it work, but it wouldn't. Far too complex, expensive. I didn't even mention the vaccines in dry form, in microparticles, and they're coated on gold microparticles. So helium-powered gold microparticles, the cost profile, as you can imagine, would go through the roof. But even if we got the cost profile correct, these sources of variability were far too great to make it consistent and work in Africa. And that really was confronting for me. Are many of you in the room inventors? Have you come up with patents before or are looking to? It's not much of a show of hands. Some twitches here and there. Well, it's interesting when you look at people that come up with ideas and patents, it is an analytical relationship. You do look at the science. Good inventors do this and you think analytically, but it's an emotional relationship as well. You actually want the technology to succeed. But the Gene Gun I found, no matter how hard I tried, it wouldn't work. And so in response to this, I took a step back and encouraged by my colleagues at Oxford, they said, Mark, why don't you go and map the skin's immune system? People haven't done that before. I thought, yeah, okay, I'll try that. And started learning the field of immunology. And it's another confronting activity. Immunology, historically at least, has been quite qualitative rather than quantitative. And this led to a moment in time for me, and believe it or not, the eureka moments can actually happen. Funnily enough, for me, the moment was I was sitting in a lecture, and I was a little bit bored with the presentation, started doodling, came up with an nanopatch. Now I'm looking around the room and I'm looking for people that are bored. Turns out it's kind of okay, so you have a license to invent something new if you choose to. So the idea was this. Now I don't have a laser pointer, but there is one. No, that's okay. This kind man's going to be serving a role as an assistant shortly. That's okay. I'm going to do it in a different way. I'll walk over here. So, see these circles just here? That's the skin's immune system that I mapped. They're called antigen-presenting cells. People didn't really quite know where they were before. And see those black circles just there? That's what happens with the Gene Gun when the microparticles hit the skin. Now, the thing at the top is a half cross-section of the smallest needle that you can buy. Okay, so the actual one goes off the board. So if the question is this, how do you place vaccine directly to that cell, that cell, that cell, that cell, and that cell, which is what the immunologists were telling us, a needle wouldn't work because the scale is just wrong and the target is too shallow. It just wouldn't work at all. The gene gun, it does happen from time to time, but there's a lot of variability and it was very difficult to control. Then I thought about what's the ideal thing if you could remove all constraints and just the dream experiment. And the dream experiment is here. Like a nano-injector, something to inject vaccine directly to the nucleus of this antigen-presenting cell. It's a fine thought experiment, but in actual fact, you can't do that. It's impossible. It's too small, and even if you did manage to pull that off, you'd need to inject hundreds or thousands of these cells. But then I thought, well, okay, what's the backup position? What can we do? And I thought about this thing on the right-hand side, an array of projections, okay? And use a probability-based analysis to ensure you have a direct amount of hits. And while you're at it, dry coat the vaccine to the tips of these projections so the vaccine is dry and doesn't need refrigeration as well. That way, you have improved vaccination by improved immune responses. It's needle-free and you remove the cold chain. So that was the moment. That, by the way, was a little while ago. It was in 2003, 2004. I returned to the University of Queensland in 2006 as a professor of biomedical engineering, and my mission was quite simple, but not easy. And the mission was to take the nanopatch forward and try to make a difference in this space to turn it into something real in healthcare. First step was to make it work. How do you make these nanopatches? How to invent a new way of doing that? Now, we're going to try something here. So this is an example of a nanopatch. This is a silicon wafer, and we needed to use deep reactive ion etching to make it. And yeah, we can have a go at this. So this is, please welcome my assistant. There it is. So the short answer is yes, we could make it. It's a small chip. So instead of using rockets to go into vaccines, it was taking silicon chips to go into vaccines. So another form of field hopping. And that particular case there is a device for human use. And under a microscope, you'd see 10,000 tiny little projections for that probability-based analysis I was telling you about before. Thank you. Okay, so that worked. That's great, thank you. So yes, we could make these nanopatches, but do they actually work? And what does work mean? So this is a close-up of one example of a nanopatch that I designed for mouse work. There's 3,364 projections there, and you need to dry coat vaccines to that. But you can see it compared to the needle and syringe. There's nothing sharp to see. When we take these nanopatches, these devices, when we push them into the skin, do they go into the sites? I'd like them to. Here's an example of data that we generated with a scanning electron micrograph. And although it looks like a cartoon because it's got false color there, it's real data. We have the tough outer layer of the skin, which is red. The next layer of the brown is the viable dermis, and this purple color is the dermis. And we haven't stained here for those particular cells, but I'll tell you about them in a moment. But yes, the projections go in reasonably consistently. But that didn't happen by accident. There was a lot of work. We had to understand the mechanical properties of the skin. And I won't go into all the detail of how we did that. These are just some examples of strain rate dependencies and also scale dependencies that we needed to understand because no one had measured it before, the mechanical properties of the skin. So that's one piece of work. And another, when we look down on the skin, you can see what's generated once we apply the nanopatch to the skin. So yes, they do engage with the skin. Yes, they can be made. But do they improve the way vaccines work? Well, to test that, we had to first find a way to coat vaccines to the tips of these projections. And that's a pretty difficult thing to do, and I'll explain it just here. So although these things look really big just here, in actual fact, you can see the scale bar there. It's about 40 microns. So we are dealing with structures that you can't see with the naked eye, and our job was to dry coat vaccines to the tips of those projections that we can't even see. And we needed to invent new ways to do that. And this is one example of dip coating that we pioneered. There's a few other methods, and there's other methods that we've taken forward, including a gas jet or a bubble jet kind of methodology. Do the vaccines actually go to the skin to where we want them to? So this was an important experiment, and I'll set the scene before I ask the guys in the back room to press play. We're using a multiphoton microscope here. Those cells that I was telling you about before, the green ones, are antigen-presenting cells. We've stained there with what's called an MHC class II marker. And it was a fun instrument to work with, this multiphoton microscope. And the blue are the nuclei of those cells, and the red is the vaccine that was delivered with these projections from the nanopatch. Now, I'll see if we can ask them to press play, or maybe I can. We'll see. Okay, great. So first up, you can see there's an array of red, and then if we zoom up just there, you can see it's in contact with the immune cell. In fact, it appears as though the dendrite of those cells is wrapping itself around the vaccine that's being delivered. So that was quite exciting and lined up with a probability-based analysis. We assumed there would be 2% of the cells that would be targeted by the nanopatch projections, the vaccine delivered, and that turned out to be the case. And I was very excited. But it doesn't really prove that it'll make a vaccine work better. It's still not the important experiment. So the only way to find out in the end with vaccines is you need to vaccinate. And we're working with the animal model, and so you'd vaccinate with, in this particular case, an influenza vaccine, and then wait. And it's a terrible waiting game. You're waiting for a month, sometimes 2 months, in many cases longer. And that's what we did. And this is an example of what we found. And let me set the scene on these data. So this is 1 month after vaccination, a standard influenza vaccine. It's a split virus vaccine. And on the horizontal axis, we have the dose of the vaccine. On the vertical axis, we have the immune response. Now, the immune response is a parameter called the hemagglutinin inhibition titer. And the dashed line is the accepted wisdom of what's considered a protective. So if you're above that dashed line, it's working. And if you're below the dashed line, it's not necessarily working. And the standard dose with the needle and syringe is over there on the right-hand side, 6,000 nanograms, 6 micrograms. You deliver that, you get a protective response, and that's great. The blue line, though, is what we generated with the nanopatch, and we found a vastly different set of parameters. Here's one way to look at it. So take a look at this condition just here. The vaccine wasn't working at all with a 60 nanogram dose, while 60 nanograms delivered with the nanopatch was protective. And we explored this further. We repeated it and we repeated it. And this is in the animal model, and we found that around 100-fold dose sparing was achieved compared to the needle and syringe. That's important. It's important particularly if you're dealing with expensive vaccines that are very difficult to make, like the HPV vaccine, the human papillomavirus vaccine, and you're trying to improve the reach of vaccines into low-resource settings. And it's a big problem. There's 14 million deaths per year due to infectious disease. And one of the key problems is the cost profile of those vaccines and making them work better for the developing world. Now, I'll show an image that might look familiar to some of you. So this is just one snapshot of the challenges in maintaining the cold chain with vaccines. So the vaccine needs to be refrigerated in many cases, and the tolerance for that refrigeration is quite tight. You can have a vaccine that's too cold. So you need specially dedicated fridges, refrigerators, I should say, to keep the vaccine at the temperature it needs to be. And when you're in resource challenge settings, like say in a desert, you need devices like this for that last mile of transporting vaccines, and that's been one of the key challenges. Now, because the vaccine is dry, coated to the nanopatch, the thinking behind that is it doesn't need refrigeration. It's to do with how the chemistry works, the interactions are locked in and slowed down versus what they would be like if they were in liquid form. And it came to pass in this particular case. Here we have a trivalent influenza vaccine, and we coated the nanopatch, stored it for 12 months versus freshly coated devices, and didn't see any loss in how effective the vaccine was, even though it was stored at 23 degrees Celsius. So thermostability was maintained. So what's the next step in the journey? It's all well and good to have vaccines working in the mouse model and in the benchtop. It was to take it forward and get it into humans, and also, while you're at it, turn it into products. And so in 2011, I founded a company called Vaxxus and helped take that forward. And the driver for that was this work, this kind of thinking. Now, I was fortunate enough in 2012 to win a thing called a Rolex Award for Enterprise, and they provided 100,000 Swiss francs that I was able to put to work and for the very first time get into the field for developing world use. And this is a neighbour of ours in Australia, but for most people is a very long way away. It's Papua New Guinea, which is one of the, it's a resource-challenged location. And one of the problems there is vaccinations are too expensive and the terrain is difficult for transportation. And so we did a usability study of the nanopatch, and the success of that particular study laid the groundwork for clinical trials that have taken place and funding from Bill and Melinda Gates Foundation and the World Health Organization for polio eradication. And the nanopatch has now been taken forward into the clinic and shown to be clinically successful, and it's on the commercial pathway going forward to become a product. So that's exciting. But I want to pause just here because my job today was to talk a little bit about what's coming with the fourth industrial revolution. And yes, the nanopatch is an exciting device, and the gene gun is and was an exciting device too. But I see those as both embodiments of the third industrial revolution, and also in the world of classic pharmaceutical science. And it's a slow game. It's a difficult game, don't get me wrong, but in the pharmaceutical industry, to go from idea to product is usually about 15 years, and that's when everything succeeds. So let's talk about healthcare and the fourth industrial revolution. Now, I've touched on this very briefly. I talked about different fields, and you've heard this from other presenters, phenomics, genomics, all manner of things taking off, mathematical analyses. I heard the other day that we can all be described as a parabola, which I found really exciting. What I want to do is lay a bit of groundwork for one stream of what's coming, I think, and that stream is, it's this. So we're all familiar with wearable devices, and some of us may be wearing things like Apple Watches and so on. And they've come out of nowhere. The Apple Watch was only released a few years ago, the very first generation. And they're really interesting and good devices, but they're not functional medical devices properly just yet. But what is happening in front of our eyes is there's a march up the value chain that's taking place, where genuine medical device wearables will be there. And the question is, well, what problem are they going to solve? Why are we even doing this? So I've talked about infectious disease before, and that's a massive problem. But the biggest global healthcare killer at this stage in human existence, at least, is cardiovascular disease. There's about 20 million deaths per year due to cardiovascular disease. And there's a subset of that, which is heart attacks or myocardial infarction. About 7 million deaths due to that event. Now, there's a lot of reasons why those heart attacks take place, but it's a kind of thing that if you had a wearable device that could detect the early onset of a heart attack, then you could buy time to allow the intervention to take place, to prevent that from happening. And that's an important problem. And that's something that we're working on. Coincidentally, this new thing that we've put together called WEP2020 is backed by the Australian National University, and our job is to work in this particular space. And the devices that we work on are called micro-wearables. If I touch on the science of this, so let's talk about the skin for a moment. The good news is that the skin is an amazing organ that does its job. And the job is to keep the bad stuff out and the good stuff in. Now, typically for drug delivery, you need to breach that barrier to get things in, but now let's talk about the other side, which is sensing. Now, just a little side anecdote that I want to touch upon, it's this. As an engineer, we can see that the average car, like a Toyota Corolla, has 200 sensors in it, okay? Yet, we're not instrumented at all. So why can't we kind of apply some of that smarts of engineering into medicine where we can gain access to all manner of helpful signals? So that's one of the drivers. Now, I mentioned the skin being an effective barrier, keeping the bad stuff out, the good stuff in. Well, today's wearables are surface-based, so they're on the surface of the skin, and the most important signals are typically deeper within the skin. Blood-borne biomarkers is one example. So trying to gain access to those from the surface of the skin is kind of like standing in one room and there's a wall and trying to listen into the next room. Now, depending on the thickness of the wall, of course, but you don't really hear much of what's taking place next door, and what you do hear is muffled. And that's kind of like what today's surface-based wearables are like an Apple Watch. If you really do want to gain access to the signal, another way of doing this is put a hole in the wall. Now, that's an example of an implantable device. Yes, you can put that device in and gain access to all manner of signals, that's for sure. But it's a surgical procedure, and that device is within the body. You might need to take it out at some point in time. You have biofouling, you have all manner of issues to work through. What we're doing is concentrating on, I'm going to be a little bit short here, I think, this layer just here, the viable epidermis and the upper dermis. And it turns out that particular layer, if you can access that with functional stealth-like medical devices, you can gain all of the, or most of the upsides of an implantable device, but it would feel like a surface-based wearable. And so that's the core concept of what we're about with micro-wearables. So another way of looking at this is there's a couple of embodiments that we're thinking about and working on. We're doing more than thinking about it, we're working on it. And one is a hydration device and the other is a cardiovascular device. So the second one, as I mentioned before, continuous monitoring of a biomarker like troponin to pick up the early onset of a heart attack. But I thought we'd do something a little bit different today, so we're going to do a live demonstration. So I need to call up my assistant. Thank you. Can we welcome him again? Now, I want to set the scene here. We haven't done this before publicly, so you're in a very special crowd here. So we have a thing called, it's a hydration micro-wearable, okay? And I'll grab that device in a moment, but we're having it talk to this particular app just here, but I'll just have it connect in a moment, excuse me. So what you'll see in a moment is, first up I'm turning this device on, so it's flashing. And I'll make sure it's paired. Okay, looks like it's paired just now. So if you can put that underneath there. Oh, that's it. No worries, I'll get this. Okay, so if we hold that under, so, oh, there it is, great, so that's working. So there's no signal at the moment, so go ahead, you can hold that. Okay, so what I'm going to do is take this device and apply it against the skin, and the micro-wearable will go in, and you can see it's measuring my hydration by targeting the viable epidermis. And the good news is it's within target range. It means I don't need to drink anything, and I'm not dehydrated. But that's real-time monitoring of hydration, minimally invasive with a tiny little square that goes into the skin, just a hair's width in, a stealth-like device. We take that off, the signal disappears, and hydration has a big medical need. If you're elderly, for instance, you can forget to drink. There's sports utility as well. There's many different things that we can tackle with that one particular device. Another example of an embodiment that's a stripped-down version of the one that I just showed you is this one just here. I'm not sure if you can see it. There we are. Okay. And what that is a sports device, continuous monitoring of hydration. So if you're a tennis player, for instance, or an elite athlete, say in a cycling context, continuous reading of hydration to tell you what you're doing. Another context that's really been exposed to us within Australia is firefighting. So firefighters, one of the key risks is dehydration, and these devices we think could really make a difference within that space. That's just, what I've just presented is just one little keyhole of what might be coming by gaining access to data, by using smart science, good engineering, and having a great team. And our team in Optimo is pretty much the best team I've ever, actually it is the best team I've ever had the opportunity to work with. We've got people with PhDs from Oxford, Cambridge, Stanford, Boston University, and other great universities. And they come from all walks of life. We have biologists, engineers, chemists, and we're applying what's called a skunkworks model. So we're not an academic group, but we're doing it a bit differently. We are a company, but we're applying this model to allow us to go quickly, to come up with these kind of healthcare outcomes as quickly as possible, because we believe time matters. So in the interest of time, I'd like to finish with one last slide, which is nothing. And I've done that on purpose. So that's a blank canvas for people here in the room to look to paint, if you like. So the fourth industrial revolution, we don't know who the champions of that will be. I really believe there's a great role to be played for engineers and scientists to be brave enough to field hop, to go into working medical problems. It's not easy to do, but it's really worthwhile doing it and make a contribution within this space. And who knows, one of these faces that I'm looking at today might be occupying this picture in the future, and I hope it does. So with no more ado, I'd like to thank you for your attention. If we have time, I'm happy to take questions. This one?
Hi, thank you for amazing talk. I'm really excited with this idea of wearable device. I was wondering how long these sensors that you test with Professor Halliday last, and how often can you have new measurements?
Yeah, so as an engineer, I'm a firm believer of walking before you run. So keeping it simple, yeah. So the first embodiments of these devices, I think, will be single time points, in spite of what I've just showed you, because that's the simplest thing to do. So like in an ambulance setting, getting a readout immediately to tell you that you're having a heart attack, that can be very, very powerful. That's the fastest way of getting it out there into the public. How long they can work for, we'll see. My knowledge of dermatology suggests that maybe about 3 weeks might be an upper limit, because that's the sloughing off time, the turnover of skin cells tends to be. But we've not explored that parameter just yet.
Thank you.
Yes?
Hello, thank you for your presentation. I had actually a question about the dry vaccination method you showed, which is very cool. I was curious about two things here. How expensive is it to make these, well, stamp-like things? And on the other hand, can you reuse them?
Yep.
And how would you do this?
So you're asking about, I think you're asking about the nanopatch?
Yes.
Yeah. So the manufacturing method suggests it could be well less than $1 per device, the device itself. So it was important from the very beginning for it to be competitive against the needle and syringe. And the needle and syringe tends to be about 7 to 10 cents per device. So it's more expensive, but if you only need a fraction of the dose, you don't need necessarily a trained practitioner to remove the cold chain, you can knock it out in price in other areas. I think there was another component to your question about the stealth-like devices.
No, I mean for this micro patch, can you reuse them, and how could you achieve that if it's not possible yet?
So you're talking about the micro wearable or the delivery patch?
The vaccination methods.
No. So it's designed to be single-use disposable. You want it to be that. So You want to avoid situations that present risk for cross-contamination between patients. Yep.
Thank you for the great talk. So, for the same devices that he was asking about, so you're drying it up, so you would also need to control the humidity, isn't it? So, otherwise, it will again functionalize, and I mean, the same issue will happen.
That's right. So they're stored, the devices are stored in packaging with controlled humidity. So you take it out of the packaging, then apply it. And the kinetics of, even if you're in a very humid environment, the kinetics of how that's being affected, it'll take a very long time for the vaccine properties to change. So that you have that time window from packaging to application.
Great, thanks for the very great talk. I just want to ask, like, how simple, I mean, to commercialize research is that from your own experience, you open up companies and everything, and as a scientist, as engineers, and also as students, I mean, like, I can see a lot of problems, and we can provide solution, but somehow the solution just become isolated, but because we don't know how to commercialize it. Maybe you can share some of your tips and secrets. Thank you.
Oh, look, I'll try. So I was looking at Kevin over here because I think Kevin will have views on this as well. Yeah, it's not easy. But most stuff isn't easy. So science isn't easy. Research is not easy. Doing a PhD is not easy. So it really comes down to, let me think about it. So I think only try to take it forward to turn it into something real if you have the belief and the passion to do it. If you're trying to do it just to tick a box because you're seen to be commercializing, it's not going to work like that. You have to really feel a sense of purpose tied in with it. I think that's really important. Determination ties in with that. There's a lot. You have to work through a lot of no's to get a yes. So being smart about it too. So if you are getting the no's, learn from those. So fundraising is an example. So my previous company, it took 70 pitches to do the fundraising. You think, oh, that sounds terrible. But it actually wasn't, because each one was a conversation where you'd learn, and you're actually updating your narrative as well from that. So it's having that open mind on that. And I think also working hard to not fall into being in a certain tribe, yeah? So what tends to happen is that you have the commercial team tribe, and we have the academic team tribe, and we all throw stones at each other. So the academic tribe will say, oh, it's just engineering. Once we've done it, it's easy. And the commercial team will say, well, you know, the academics are in la la land, right? So, and that's a direct quote. It wasn't from me. So if you have the ability to work, work across that and have one work with the other, then you can really create something special, I think. I'm sure there's other parts to it, but there's some initial reactions to your question. Kevin, did you want to add anything to that? There's a microphone.
Yeah, thank you for that hospital pass. I'm not going to suggest that commercialization of technology is easy, because it's not. But certainly one of the things we're doing here at KAUST is to take away some of the pain points, to make it as simple and as straightforward as possible. I mean, over the years, I've watched academics struggle to have to try and learn some aspects, techniques, patenting and everything else. Really, they don't need to. And our approach here at KAUST is to try and make sure that the academic is doing the thing that the academic is really good at, and not having to do things that they don't necessarily need to do. So I'll repeat, not simple, not straightforward, but if you've got a passion to do it, like this gentleman here has, then we can absolutely help you to make the difference that you want to make.
Thank you. Because for me as a PhD student, the one thing that I know to commercialize my research is by publication. That's it.
Come and see me. Come and see me.
All right, sure. Thank you.
Yeah, publication is important. It gives you your credentials and your platform that you can build upon if you have that passion to take it forward. Yeah.
Yeah, thank you for the nice talk. My question is regarding coating of these microneedles with the vaccine. So is it true for all type of vaccines? Because each vaccine has a different chemistry, different structure.
Yeah.
So when you coat them, they might, like behave different. So, so did you find that pattern, or is it same for all type of vaccines?
Yeah, great question. So all vaccines are not created equal, right? So what we found is we used excipients that's dominated the vaccine. So it became an excipient. So an excipient is just a thing you add into the mixture. So methylcellulose is one example, is carboxymethylcellulose. And it really became a question of the behaviour of the excipient rather than the vaccine itself. Having said that, there's a thing called an adjuvant with the vaccine. So this is something you add in to the vaccine to make it work better. An analogy for it is it's like a turbocharger on an engine, okay, for those who like engines like I do. So, but some adjuvants are oil-based, and there's one example, it's called alum. It's an aluminium hydroxide, it's oil-based, and it's not really well suited to a lot of coating methods. So that's one challenge in the mix.
Okay. I just want to remind all the biologists and biochemists here that what we heard this evening was a rocket scientist telling us that biology is really, really hard. As a chemist, I'm terrified of biology. It's far too difficult. Thank you. Thank you. Mind officially blown. Thank you for coming. We're going to ask you to stay up here for some photographs, but please join with me in, again, thanking Professor Mark Kendall.
Thank you. Thanks, Kevin.